Good evening, everyone. I'm Robin Garrell, the president of the CUNY Graduate Center, and it's a pleasure to welcome you to tonight's event, which is part of our City of Science series of public conversations here at the CUNY Graduate Center and our Advanced Science Research Center, which is located in Harlem. The City of Science series was started in 2017, and we've presented more than 24 events. Our goals are to bring a deeper understanding of science to the general public, to raise the profile of sciences at CUNY, and to provide a platform for the work being done by CUNY scientists. Since its inception, the City of Science program has been led by Josh Brumberg, a professor here, a Dean of Sciences at the Graduate Center, and interim director of the ASRC. And he works in partnership with Karen Sander in our Office of Public Programs. So thank you very much, Josh and Karen, for your leadership. Tonight's event and the entire series represent the kind of programming that makes the Graduate Center a crossroads for discourse and discovery here in the heart of New York City. It's great to be here in person. It's one of our first public events here at the Graduate Center since 2022. And it's even better than it was then because now we can engage a global audience. Since the spring of 2020, the Grad Center has live-streamed events that have reached audiences in all 50 states and more than 40 countries. So whether you're with us here today at 5th Avenue and 34th Street or somewhere else in the world, welcome. I'd like to extend a special thanks to our partner in presenting this conversation, and that's the BGI Nobel Laureate Archives Program at Cold Spring Harbor Laboratory. The laboratory is a powerhouse for biomedical research and education and is the professional home to 8 Nobel laureates, and we are honored by this collaboration. Tonight, we will listen to a dialogue between 2 extraordinarily accomplished and visionary scientists, Professor Phillip Sharp and Siddhartha Mukherjee. You'll hear more about them and their seminal discoveries in just a moment, but I want to highlight the fact they have both demonstrated a commitment to bringing their scholarship to the public, helping all of us understand science and health in new and interesting ways. And at the Graduate Center, we embrace this approach, encouraging our students and faculty to find opportunities to make their research relevant and accessible to general audiences. Promoting research that advances the public good is core to our mission, and we're so pleased to advance that purpose by hosting the conversation this evening. Now, as a chemist and bioengineer myself, I'm especially excited to get to know our 2 very special guests better. Finally, a word about tonight's format. Professor Mukherjee and Professor Sharp will converse for about 40 minutes or so. Then we'll have a chance to take some of your questions. So, this is to the members of the in-person audience. I invite you to write your question, just 1 per card, please, on the index card they were handed out when you came into the auditorium. The ushers will collect them right around 7 o'clock or a minute afterwards. So have that in mind, so you have your question written out, and we'll collect them and feed them to our speakers. So now I'm pleased to turn the mic over to John Tuke, the head operating officer of the Cold Spring Harbor Laboratory.
Thank you very much, Robin, for such a warm welcome. And I could not agree more with you that being able to gather here in person and benefiting from the presence of our virtual audience is really exciting. On behalf of Cold Spring Harbor Labs, I'm also delighted to welcome everyone, whether here in person or online, to the beginning of what we hope will be a recurring collaboration between Cold Spring Harbor Lab and CUNY to host an event in conjunction with our BGI Nobel Laureates Archive program. We want to thank BGI for this support and acknowledge Henry Yang and the founders of BGI, and welcome Charles Bao from BGI Americas, who's with us here tonight. I also want to highlight our ongoing collaboration with CUNY at City Tech and the DNA Learning Center in Brooklyn, for which we are immensely proud. That Nobel Laureates program is one of several initiatives through CHL's Center for Humanities and History of Modern Biology, which also conducts oral history interviews with scientists, hosts conferences on the history of science, and offers fellowships to visiting historians, filmmakers, writers, and artists. The center is located at our historic library and archives, which documents not only 130 years of research and discovery at Cold Spring Harbor Lab, but also holds the manuscripts of amazing scientists from around the world, including Francis Crick, Carol Greider, Sidney Brenner, and New York City's own Evelyn Witkin. The center, as well as the library and archives, is led by Mila Palk, who helped make tonight's event happen. She's under the weather and asked that I share her remarks and represent Cold Spring Harbor here tonight. Echoing Robin's comments, we are deeply honored to be joined by Phil Sharp and Sid Mukherjee. Phil Sharp is a professor at MIT, author of several hundred scientific papers, and winner of the most prestigious honors and awards in biology. That includes the Nobel Prize in 1993 for his discovery of split genes and the NRA splicing mechanism that edits out the intervening segments. That discovery of those nonsense segments that are edited out is very important in his career to cancer research. Phil has devoted much of his career to cancer research, along with co-founding the therapeutics company Alnylam and the biotech company Biogen. He is one of America's most distinguished scientists, and we are thrilled to have him with us here tonight. Sharing the conversation with Phil is Professor Mukherjee. He is a cancer physician and researcher at Columbia University, as well as a prolific and accomplished author. He won the Pulitzer Prize for his 2010 book about cancer, The Emperor of All Maladies, A Biography of Cancer. You may also have seen the Ken Burns documentary based on that book, or another one based upon his 2016 book, The Gene in Intimate History. Next month, he will have a new book out, The Song of the Cell. I was trained as a historian, and I've read the first two, and I'm really looking forward to the third. So on behalf of the lab, I am so grateful to Phil and Sid for joining us here tonight, and it's now my pleasure to turn things over to the two of them to start the formal programming.
Thank you very much, John. A couple of quick errands when I start programs. Would you please all turn your cell phones off? Make sure they're off. Thank you. So Phil, in knowing you and reading about you, and we've known each other for many years, you resemble the molecule that you study. People say that, people say that humans begin to resemble, or dogs begin to resemble their owners. Sometimes scientists begin to resemble the molecules that they study. And by that I mean something very special, which is that your discoveries have been as diverse and as prolific and as important as RNA, the molecule that you spent most of your life on. So there's so much to cover that I thought I'd like to give people a roadmap of where we're going so that you know sort of what the milestones are as we cross them. We'll talk a little bit about your biography, early days, leading mainly up to your postdoc at Caltech. We'll then talk about the discovery, where we'll spend quite some time, and try to demonstrate the hows and whys. Why was it so important? How it was so important. We'll then spend a little bit of time on what I call the post-discovery life, because you went on to make other discoveries. People think about, obviously people who know Phil know this, but sometimes people forget that in fact it's not just RNA splicing that was the center of your work, that there was a lot of biochemistry that followed and the applicability that followed. And we'll end with what I call applicability and medicine. How does this all become? How do you take all of this stuff, which seems so arcane, or seems so arcane, and so biological, biochemical, and make medicines out of it that are transforming human lives? So that's the overall path we'll follow, if that's okay with you.
It sounds fascinating. I was worried about what you were going to use as an analogy with RNA. It could have been many things, including very wiggly.
So let's start with a little bit of biography. Just a few minutes. Walk us through sort of your early days, where you came from, and then walk us through to the time you ended up at Caltech.
So let's go back to the earliest day. I was born on D-Day in '44, and this was in Kentucky on actually on a tenant farm. My father was at that stage a tenant farmer, and my mother was engaged that day. But then I grew up in Pendleton County, Kentucky. It was a school I graduated with 60 in the class from the whole county. Played basketball, loved sports. And, but I was uncomfortable. The biggest issue, well, let's phrase it this, is the biggest issue of coming from that background is you don't know what the world is like, and you don't know how you will be accepted in that world. And so I went to a small college. I paid for most of it from work I did on the farm. My parents gave me the money from it, and I used that, took a loan, went to a small college in the mountains of Kentucky called Union College, fell in love with math and chemistry, ran into a professor named Dan Foote, who just came from the University of Illinois, applied to Illinois for graduate studies. I completed my degree there in about 3 years, doing theoretical work on polymers. And DNA was one of those polymers. And while I was there, I fell in, really was attracted to the work of a professor at Caltech named Norman Davison. And I decided that, and I had been reading books about molecular biology and DNA, and I decided at that moment that I wanted to apply my biophysical and chemical understanding to this molecule, DNA. And this was in '69. And I went to Caltech with Norman Davison, who was also a biophysical chemist who was moving into DNA, and Ron Davis, who you know was a grad student with him, first to map a gene on a genome. And so I launched into molecular biology at Caltech, a very special place. And then it evolved from there to Cold Spring Harbor Labs, where I was at for 3 years. And then in '74, I moved from Cold Spring Harbor Labs to MIT.
And so, you know, this is a time, I'll talk a little bit about Norm Davison in a second, because kind of an important figure, pivotal figure in many people's lives, unfortunately forgotten by many, but a very important figure in many people's lives. But connect that for us from DNA to RNA, as it were. So here we have DNA, the queen of all molecules, the queen of all biological molecules. And you go to Caltech, Cold Spring Harbor, MIT, and at some point of time, your research emphasis, and you studied polymers, you studied chemistry, you've done a lot of biochemistry, you're in Norm's lab, now setting up your own, and your interests begin perhaps to shift from the queen of all molecules to RNA, the handmaiden, that then becomes all of a sudden the new queen. So tell us about that shift. Tell us about what it takes. I mean, you're a young professor with, 4 or 5 people in your lab, 3 people in your lab. What does it take? And walk us through that intellectual moment in which you said, I'm really going to study RNA and its metabolism and its properties.
So let's phrase the journey this way. When I was at Caltech, the science of DNA and RNA was a science best illustrated by this.
You brought props.
I brought props. What you see here is a rope, and actually DNA is almost rope-like when you look at it at microscopic scales. And we knew at Caltech that in bacteria, the gene looked like this blue bar. It was a defined length. You copied RNA from it. The RNA was then translated to protein. And there were sequences near the gene that typically would turn the gene on and off, depending on external conditions. So you had regulation. So we couldn't isolate genes at that stage. That came a few years later. But by genetic manipulation and other manipulation, we knew that this was the structure of genes. And this was all done in bacteria. It was very sophisticated. Bacteria are wonderful because they grow so fast. You can mutagenize them and not pick mutants, study variants. Variants tell you a lot about what the normal process is because you can perturb a gene and ask its function. But I was interested in what human genes were like. I thought the science that we had in manipulating DNA and mapping DNA on chromosomes, much of which Norman did, and Ron Davis, that there were hints that genes and RNA was different in our cells. And that puzzled me. It was...
Tell us, what years are we in?
We're talking about 66 to 69, 69 to 71.
Okay.
So we couldn't isolate human genes. The chromosomes are too large. We had no tools to really map them. And this was at the time we were talking, or the technology to do recombinant DNA was just appearing. And there was these hints in mammalian cells, much of which came from faculty professors like Jim Darnell at Rockefeller, that there was an unusual long RNA in the chromosomes of, in the nucleus of human cells that would be inconsistent with this gene structure. But only a few people paid any attention to that data. But it was really a puzzle, and to me, it sort of begged the answer. Is there a new set of rules in human molecular and cell biology? And that's what led me from Caltech, because I did 2 years at Caltech studying bacterial genes, mapping them, wonderful science. But I wanted to make the transition to looking at human chromosomes and human genes.
Great. I have a personal anecdote about this, but I'll keep that prop because I'm going to ask you to do something in a second. So I studied with Paul Berg. I was studying in the back. I studied with Paul Berg, and Paul was also awarded the Nobel Prize for discovering recombinant DNA. And Paul always has given this lecture. He was studying viruses, human viruses, a virus called SV40 in particular. And he, too, was puzzled by the fact that even though, just to put it very simply, that something about the genetic structure and the structure of the RNA and the protein that came out of it did not make sense. There had to be some difference between the way the gene was organized and the RNA that came out of it and the protein that ultimately came out of it. And he said, I just missed it. But you didn't miss it. You actually, you figured that piece out. So what I want you to do, I'm going to hold the mic now so that you can use both your hands. And I want you to show us what exactly happens. What is that missing piece that you discovered? Use your prop and show us what
I
that
have
missing.
another prop.
Okay, use your other prop. So I'll hold the mic. I'll come close to you. Hold the mic so that you can use both your hands, because you'll see why both hands are necessary, probably.
I want to start here. Paul Berg was making deletions in the middle of a gene, and he conceived of the gene this way. And he made those deletions, which means he removed DNA sequences. And as I said, this encodes a protein, and what you would expect is the protein would get smaller. But it didn't. And he was puzzled.
I mean, this is really puzzling. So you have DNA, you have an RNA piece, and you have a protein. It should.
Central dogma.
Yes, a central dogma. But so that's the puzzle. Okay, let me emphasize, this is a big puzzle. This is not like one of those small itty-bitty puzzles. So on we go.
Well, genes are big business because you have about 20,000 of them, and that's what makes you work. Well, folks, this is what your genes look like. Now, what is this? This is DNA equivalent from your cells. And you see these patches of blue? Those patches of blue, all three of them, code for the same protein. But in the middle of those sequences are sequences that aren't blue. They're yellow here. They're called introns, and they're nonsense, by and large. So you can't make the protein without removing those sequences. And that immediately told me that the RNA from the gene was spliced. And what does that mean? That means that the RNA is made from the genome, collinear with this sequence, and then it is spliced out. Let me turn it this way. Well, let me make it this way. Then it's spliced out so that you see removal of those sequences give you a continuous blue. And therefore, you can make the protein. And the message is this blue product of the splicing reaction. So what Paul Berg was doing was making deletions in the intron of that gene in the virus, SB40. And that didn't change the size of the protein because
It's just cut away.
it was just spliced out by a process called RNA splicing. Now, this is simple. The typical gene in your body has 10 of these introns. There are some of them that have 100. And I have another rope, which I didn't bring, which shows you the dystrophin gene. And I was giving a big lecture at MIT, and I spent a whole night making that model. And it has 77 exons in it, 77. And it's a big rope, it goes across the stage. And I spread it out in front of the lecture hall, and no one could see the blue. There were such small amounts of blue and enormous introns that you couldn't even see the blue. So you have genes that are code for one protein that's 1 million base pairs long, enormous, the size of a whole bacterial genome. Now, well, you say, well, that doesn't mean, what's interesting about that? You just splice out these introns, and where did they come? And we can talk for hours about that. But what happens in many of your cells, where we have 10 of these exons, is that they're not all spliced in the order that are along the genome. In some cases, These two exons get spliced together, and that one gets discarded. And therefore, you have one gene that makes two different proteins. In fact, almost all your genes undergo this alternative splicing, and some of them make thousands of different proteins. So even though I say you have 20,000 genes, you have the capacity through this alternative splicing, which varies from cell to cell in your body, to make many other combinations of proteins. And believe me, even though we have the genome sequence, no one can tell you exactly in one cell in your brain how that DNA is being expressed. So we're just getting the technology to begin to do that now. Now there are mutations that cause mis-splicing. There are conditions that shift the splicing. So you can't understand human physiology without understanding the structure.
Great. The next time we'll bring you the dystrophin gene, and people can stretch from one end of the auditorium to the other with little bits of blue. But so that begs the question, now we're coming back to biology, when we find a phenomenon, the first two questions we ask are why and how, and the third question we ask is what do we do with it? So talk a little, we already started about why. So why does this process happen? I mean, it seems like a massive waste of energy to make this giant RNA product and then throw away 90% of it. Why does it happen?
Why does this happen? It's still, philosophers and scientists are still debating. But I think the most general and probably for certain component of it is that we're not selected at the cellular level or any multicellular animal for efficiency. We're selected for complexity and the ability to evolve. And by this alternative...
That's a very, very interesting statement, right? So go ahead, yes, keep going.
So by using this flexibility of alternative splicing and using the same piece of a gene, many of these exons, their related cousins appear in other genes. So we created genes, we in evolution created genes by taking these exons and moving them around into other genes and having them spliced into those genes. And the other thing, so I think that's the answer for why it's there. Mm-hmm. And where did they come from? They come from, this is another story, folks, you don't know about. they come from transposable elements, DNA parasites related to viruses that long ago got in the germline of all multicellular animals, probably all bacteria as well, and they jump around and duplicate themselves. So most of these introns we can trace back to that duplicative transposition of DNA. And some of those are viruses that probably were transmitted horizontally and then got in the germ cell and became part of the DNA. So the evolutionary perspective and the sort of understanding of how transposable elements, which Barbara McClintock contributed enormously to, is critical.
So I'm going to make one comment, and then while I make that comment, ask you to start writing your questions, because it might take you some time to think of them. The one comment is that when people have looked, it turns out that the human brain is probably one of the most, is a site where an enormous amount of splicing happens. The philosophical puzzle, which we probably never would discover the answer to, is that when the human genome was sequenced, everyone was shocked by the fact that we only had so many genes.
20,000.
20,000, maybe 25,000 odd genes. And wheat have more genes, rice has more genes.
Worms have more genes.
So the question then became, how can you build an organism that can have conversations on stage out of 25,000 genes? And that's potentially one answer, that the most complex of our organs takes those 30,000, 25,000 genes and makes hundreds of thousands, maybe potentially millions of variants. So by taking out pieces of it and basically using the split system to combine, to split and vary, so that one cell and its neighboring cell can actually, from the same genome, build a completely different complement of proteins. And that's one potential answer. And it's, of course, being debated philosophically.
Yeah.
Let's move on a little bit very quickly. Give us a quick sense of, we're now going to move towards applicability in medicine. But before we do that, tell us a little bit about your post-splicing, your Nobel discovery. And then tell us a little bit about what you did. Now, you were still a relatively young professor when you made this discovery. And then there's a whole life as a scientist in front of you. So tell us about the other discoveries that came in the heels of your discovery of splicing.
So I was 33 when I
33, okay.
when I made the discovery.
Yeah, most people are watching TikTok.
And I had a lab of about 5 to 6 investigators. Believe me, everybody in science within a month around the world understood
Yes.
what this meant. So the whole field came rushing to this new frontier because they knew the rules we had worked out so clearly, they had worked out so clearly in bacteria was going to be different. And so I decided at that stage that what I really wanted to do was to understand the biochemistry of how those sequences were removed, because I thought it would tell us how they change between cells and something about their evolutionary origin. So I developed the biochemistry, influenced by your collaborator, Jim
And yeah,
Manley.
we've done some work on this ourselves.
He was at MIT in a postdoc, not in my lab, in Malcolm Gefter's lab. And so we started working on the transcription, how the RNA is made from that gene, and then with other people discovered a biochemical system that would remove the intron, and that was the spliceosome. And the spliceosome is a very big, complicated machine. It's been 40 years, about 40 years exactly, the structure of the spliceosome was known. So we know it now in a chemical sense. And we know that you can drug the spliceosome and you can cure diseases or ameliorate diseases with it. And there's a lot of people working on that. And we know the nature of mutations that control this process. So all those were pieces of very fundamental science that affects every cell, almost every eukaryotic organism, and is the backbone of
Right.
foundation of what you have to understand now.
Just to emphasize, I came into this splicing field because the disease that I study and treat is a disease where the main mutation is in one of the components of the spliceosome. And in fact, Jim Manley and I have been fortunate to discover one piece of it, which wasn't even known before. We are still discovering parts of that machine. But I just want you to imagine this, that just to remove that piece of DNA, an enormous
RNA.
remove that piece of RNA, an enormous molecular machine has to be assembled in the right place. And if it doesn't get assembled in the right place, you get all sorts of diseases, including cancer. Yeah. So we're now moving on towards sort of the applicability of all of this. So you, in your post discovery life, you begin to understand more and more about RNA. I know you've done a lot of work on other functions of RNA. Tell us for 2 minutes about what else does RNA do? Aside from splice itself or get spliced, what else does RNA do that we know and don't know, and what's haunting you these days?
Well, I mean, obviously, about everybody in this room has been injected with RNA, all right? So the message structure that comes out of that split gene is the structure you got injected with, all right? So there's RNA treating an infection. There's a short RNA we didn't know about this until about 2001. All your cells have a large assortment of very small RNAs in them that turn these genes up and down, on and off. And we now know how to make that synthetically and treat disease with that. We know how to follow the variation in RNA in cells and diagnose cancer and diagnose a whole host of other muscular dystrophies, spinal muscular atrophy. In fact, spinal muscular atrophy, due to Adrian Craner at Cold Spring Harbor, is treated by injecting a small oligo to an intronic sequence in the SMN1 gene, 2 gene, and it shifts the splicing. First treatment of shifting splicing. And children who had died at 2 or 3 now live.
It's an incredible thing. I don't know if you haven't seen it. I mean, the film that we made with Ken Burns follows the life of one such child. It is absolutely miraculous because you treat this child who can't walk, and it takes some time, but over time, all of a sudden, this child is trapped on a trapeze. It is just spectacular.
I mean, it made me cry when I saw it.
Yeah, it really is.
It's just beautiful.
It's incredible.
It's just beautiful.
So you've been injected with RNA to make a vaccine against COVID. Children are being treated with Spinraza, which is the drug against SMN2. We're using RNA to diagnose cancer, understand cancer. Companies are looking for mechanisms to change or affect RNA splicing. We already talked about its relevance to the diagnosis of disease. What's the question that you've taken on next in this journey?
Well, I mean, one of the things that I just want to add one more.
Please do, yeah.
One of the most important treatments of cancer now is immunotherapy, where you modulate your immune system to reject your cancer. It's the first treatment. Jim Allison got a Nobel Prize for it. It's helped a lot of people who had no other option, and they have metastatic cancer throughout their body. And in some cases, they can get cured. I think CAR T, which is designing your immune system to attack the cancer by using RNA and genes, will be the next big frontier in solid tumors.
Uh-huh.
So I think we have many things in front of us. What am I thinking about today? To get the 70 different proteins and pieces of RNA together to excise that intron requires a whole new physical process. It's an old process that we've just discovered is occurring in the nucleus of a cell, and it's called a condensate. And all these things have intrinsic disordered proteins. You don't know what those are, but that's all right. But what they do is they concentrate in the cell like a dewdrop makes the transition, moist air makes the transition to dewdrops, a transition. And that process is going to be integral to how all that regulation works. So, I mean, we're still plummeting this molecular cell biology to understand genetic diseases, chronic diseases, Alzheimer's, Parkinson's, and many of the modalities we're going to use to treat those is going to be pieces of RNA, because it interacts with this RNA that is so wiggly and all shifty in terms of how it's spliced, and we can have major effects.
And just as we end, as I said, I thought I would demonstrate the many lives of Phil Sharp. I want to just take us back in time, and then we'll start our questions. I want to talk a little bit about the whole idea that that RNA most likely precedes DNA in human and not human, but in organismal history. Yeah. So there was a world where there was no DNA and RNA, and it seems to be a fascinating world where this molecule can do all sorts of things, including behave like an enzyme, including condense with other molecules to start systems of life. Have you thought? I mean, this is not your area, but I know you follow it. Have you thought about questions about the origins of life and RNA?
Well, I think it's clear that most of the processes of genetic information transfer in biological systems had their origins in RNA-mediated processes. The splicing process is an RNA-mediated process. The chemistry that cuts the RNA is by RNA. The process by which RNA is decoded to protein is an RNA catalyzed and RNA interpreted process. And we know that very complicated viruses, polio, coronaviruses, have RNA genomes. They don't have DNA genomes. So you have a genetic system in which RNA duplicates RNA. We can do that in a test tube. Cuts RNA, splices RNA, decodes RNA. So to protein. So it makes so much sense.
Right.
That the earliest biological organism that could duplicate itself and produce offspring and carry information with it was mostly RNA. And then DNA became the storage book for genetic information when that information got very large and complex. So that's the way I view it. I think it's the most reasonable way to view it. But whether we'll ever be able to duplicate those early processes, we'll see.
Well, thank you, Philip. I mean, you're a man of so many diverse discoveries and so many diverse thoughts, and still running an incredible laboratory making wonderful discoveries. So thank you so much for being with us. And now I will turn to you for questions.
Okay. Well, thank you to all of you who have submitted questions. There's some fascinating ones here, and some springboard off of things that you just said, even though they wrote them before you said them. So I'll start with a very sophisticated question from one of our own alums, a PhD in biochemistry. Do you think that viral RNAs are older than the bacterial RNAs on the evolutionary scale? And, corollary, which are the species with the oldest splicing machinery? Or do we know?
I think the contemporary viral RNAs that we know have taken up bits and pieces of information from their host so that they primarily can replicate themselves and reproduce in tissue. The earliest virus was probably an organism that carried its own information and infected another cell and as an RNA genome. What's the earliest? Repeat the second one, because I have a better answer for that.
Over here. Which are the species with the oldest splicing machinery?
The oldest splicing machine is clearly group 1 and group 2 self-splicing parasitic agents that are RNA. They will insert themselves into a genome, and then as the RNA from that DNA is made, they'll cut themselves out.
Ooh.
They're totally parasitic and they're totally silent. And they cut themselves out by catalyzing their own excision. So that is a very old RNA.
Sounds like a really good sci-fi movie. Is there a master control of splicing, or is it all random?
Oh, it can't be random, because the information that comes from these genes has to be in a certain form, so the functional protein can then carry out what it needs to do for the cell. So we have little sequences, we're still debating all of them, using massive machine learning to try to solve it, that dictate which pieces of the RNA is removed. And we know most of that information, but what we don't know is the critical part of that information, which means why it does this in one cell and why it does that in another cell. And that's something people are working on.
And do you think AI will help us solve that?
I think AI will be a major contributor to it, because I don't think we can deal with the complexity by sheets of paper and string. So I think we have to use that methodology, which can handle large amounts of information, and then use that and work through processes.
Okay. So I'm looking at the rope, and I'm seeing that these sections of yellow they're long relative to the pieces of the good stuff, what we used to call the junk DNA. And so we're beginning to glean that there are some purposes for some parts of the yellow. What's your instinct? Do you think that actually there's all function there, we just haven't discovered it yet? Or is there just a lot of noise there amidst the signal, a lot of junk?
I'm more of there is information there, and information that directs splicing and information that directs the whole gene expression level. But I'm more, and there's lots of these transposable elements, and they drift by neutral drift, meaning you can tell in a biological organism when a nucleotide of that DNA is functional. Because then if you sequence enough genomes, it won't vary. Because if it varies, you're not functional anymore. So you can tell functional from non-functional DNA by just sequencing genomes. And we've now sequenced 1 million human genomes, essentially. So we know what is functional and what's drifting, almost random. Most of this drifts random.
Okay.
Transposable elements. So think of yourself as a Rube Goldberg cartoon. You're just good enough to be better than what came before. So you're never perfect. I can tell you that at the biochemistry level. You're just good enough.
And if we weren't viable, we wouldn't be here.
Yeah,
so.
Right.
So looking again now to the future, what do you think was the main technology advance that makes the study of RNA mainstream? So this was written in the past tense, but maybe we're looking in the future tense too. What's made RNA really a mainstream component of how we approach biomedicine and biotechnology?
I think the major technology that changed everything is the ability to sequence massive amounts of sequence. And we're now able to do that on a single cell. We can go in to a tissue, biopsy and pluck out a cell and sequence the RNA in that cell. Or you have to sequence a lot of them and then average them together, but you can then get the variation from cell to cell. So this ability to convert the RNA back into DNA and then sequence DNA from it and understand the variety of structures of proteins being made. This is only 5 or so 10 years old, is really important. It tells us about splicing and cancer. It tells us about splicing and disease. It tells us about mutations. It's just, and there's some dark matter there. We're still debating what some RNAs are doing, that we know they're there and they're very abundant and they vary from cell to cell, and we don't know what they do. So there's dark matter. I want to get over this idea that once you had the sequence of the human genome, you knew everything. Once you had the sequence of the human genome, you can start learning from it, but you don't know a lot.
It's so much work to do on the transcriptome and post-translational modification and.
Oh, the and.
So there's plenty of work to do,
you can
young
write
people.
books about it.
Well, so this is provocative. Do prions change splicing in the brain? Wow, I don't know.
Oh, yes. Prions form solids that infect cells and reproduce themselves, but in doing so, they perturb the cell's processes they could be just simple mass, but it's probably more elaborate than that. So, yes, I mean, do I know exactly how it does? Not yet. But for sure, it does.
So a topic that I think we read about in the paper a lot, which is perhaps influencing our diets, is the gut microbiome, right? So here are all these bacteria living in our guts and different parts of our guts, and they're sending signals to our brains, and they're influencing our metabolism in many ways and transcription. So does that add another order of magnitude of complexity to everything?
Yes. There are more cells living on you than cells in your body. So your skin, your gut, all your exterior-facing surfaces are an ecosystem. And they vary from you and your next individual, and they vary as you pass through life and you get various antibiotics and whatever. And they communicate. And we're just beginning to know that I have a nephew who is also in biotech, and he was working on analyzing blood with mass spec, which can identify thousands of different components. And 80% of the metabolites seen in your blood come from bacteria. So they don't have to be very abundant, but they are communicating in some way with you.
Fascinating. Professor Mukherjee, this one's for you.
Oh, goodness.
Okay. When will you be publishing your first novel? I've been waiting for it after reading the first few pages of The Emperor of All Maladies.
It's not clear that I'll publish a novel. I'm not a novel writer, but I'm interested. I think I will publish. We've gone on an incredible journey, which Phil knows a little bit about, in delving into AML. And acute myelogenous leukemia. So it's a disease that I've studied for now 20-odd years, and it's still being treated, as of today, with basically the same medicines that we used to treat it 20 years ago.
And there are very few that work. Right?
And the problem is that they really don't work. People talk about pancreatic cancer being extraordinarily lethal. This is just about as lethal as pancreatic cancer. So about 4 years ago, I had an idea about a completely new way of treating AML. And it took 4 years to get all the pieces together. In brief, the way we've been treating AML, and this goes back to your comment about cancer, when we sequenced the cancer genome, we thought that we would learn everything about cancer, and that we would then take the genome, find the mutation, find a drug, and that would be the end of the story. It's turned out to be just the opposite. It's turned out to be the beginning of the game rather than the end of the game. Some of these mutations, we don't even understand why they're occurring. So we created a new way of treating cancer, which is largely independent or does not depend on, it's mutation agnostic. And you can read about it. And it was finally turned over, the technology was turned over to a company called Vor. And so I will not write a novel, but I will write the journey of that. And it's like a novel, except it's got real characters in it.
And hopefully a very significant outcome.
That'd be great. Well, this has been a fascinating journey of life and discovery, and so many new discoveries to look forward to that have been made possible by the work that you and the students and the postdocs who've worked with you have done, who are now on their own journeys to figure out what else is in all that yellow stuff and how does it all connect. So I'd like to ask the audience to join me in thanking you very much for this evening's conversation. And thank you all for joining us here this evening at the Graduate Center. We look forward to seeing you again at future events in the City of Science and other series.